In this final short video for Chapter 7, we're going to be looking at viruses. Viruses are kind of an enigma. They don't really fit necessarily with prokaryotic cells or eukaryotic cells. They're something different altogether. In fact, we don't even necessarily consider them to be alive, at least typically. In biology, we're not considering them to be alive. And so let's get into it. Let's look at these things called viruses that we've probably heard of, but maybe we don't know that much about. Viruses are very interesting. They're not considered to be alive. They're essentially a strand of genetic material. Now, next to the genetic material, please write in that can be either DNA or RNA, can be either. So they have this genetic material inside of themselves, and they don't have any organelles. They can't make their own proteins. They can't even move on their own. But the biggest reason why they're not considered to be alive is that they cannot reproduce on their own. Remember, one of the 8 characteristics of life was capable of reproduction. Viruses are unable to do that. If there's a virus, let's say just sitting on a desktop, it's just going to sit there. It cannot reproduce on its own. It must invade a host cell in order to reproduce. And that's why, sort of classically, we don't consider viruses to be alive. Although there is some debate in the scientific community on that, at least for the purposes of my class, I want you to know that we're not going to consider them alive because they fail this test right here. They can't reproduce on their own. You can think of viruses a lot like essentially a delivery mechanism for the genetic material inside of them. So they have an outer covering of some shape and size, but on the inside, that's where they're going to have that either DNA or RNA, and the purpose of the outer structure is to infect a host cell of some type and insert that either viral DNA or RNA into the host cell, and then get that DNA inserted into the host cell for replication. Again, viruses are going to use DNA or RNA, never both. Viruses attach to the host cell using specific receptors on the plasma membrane of that host cell. So we've got our host cell membrane, you've got these receptors that are there, and the virus has these special proteins that actually attach to those receptors. You can think of it kind of like a lock and a key. And so this virus is carrying around keys that allow it to unlock specific kind of host cells. Now that then means that viruses are usually specific to one kind of host, one species if you will, and they can't just move from species to species for the most part. Not only that, but viruses, even within a species, are isolated sometimes to only one kind of cell type. For example, HIV that we're going to look at in a second, its keys only allow it to unlock one human cell, which is the human T4 white blood cell, which is a helper T cell. The flu virus only infects cells of the upper respiratory system. Some viruses might only be able to infect the liver. Some viruses might only be able to infect other parts of the body, like maybe skin cells or other epidermis cells. So it just depends on the virus. And as far as being only, usually within one species, the flu virus, which for us is an upper respiratory infection, that virus actually originates, we believe, it starts in the digestive tract of birds. So it infects their digestive tract. It usually will stay in birds. However, sometimes it will go from birds to humans. And in that case, the virus has some kind of mutation that changes the keys on its surface and allows it to infect humans. Oftentimes, that virus will actually go through mutation where it goes from the digestive tract of birds to all of a sudden being able to infect other organisms, other mammals usually, and then to humans from that mammal. So maybe you've remember the swine flu, where it went from birds to pigs to people and so forth. We'll look at this in more detail, how this works in the immune system. But for now, kind of the basics there. HIV is a very famous virus, really became well known in the 70s and then especially in the 80s. We'll talk about it in detail in the immune system unit because understanding how this virus works and how it destroys the human immune system will help us understand what the immune system is doing for us. It is an RNA virus, so it uses RNA on the inside. You'll see these outer proteins. These are the proteins that are going to allow it to actually attach to and infect the host cell. And the host cell that it affects is called the human T4 white blood cell. And we look at this kind of generally, let's give us an idea of what viruses do and how they do it. Essentially what a virus does is it attaches to that host cell, in this case the human T4 white blood cell. It inserts its viral DNA or RNA, and then that viral DNA or RNA actually then gets inserted into the host cell's DNA. And once it's in the host cell, it has a very interesting way of being able to silence all of the host genes that it doesn't want to be expressed, and instead makes copy after copy of itself and essentially turns the host cell into a virus-making factory, producing lots of copies of itself, wearing down and destroying the cell in the process. And then, of course, what are these viruses going to do? Well, they're going to go infect their own host cell, and around it goes. So that's what viruses do in a nutshell. While viruses have been a problem for much of human history, killing countless millions of us with all sorts of different nasty viruses, whether it's hepatitis or flu or measles or smallpox, scientists now are actually using viruses as a mechanism to deliver DNA. Because if you think about what viruses do, viruses get their DNA from inside of them into the host cell's genome. So the thought process is, well, why don't we just use them as a delivery mechanism? Take the viral DNA out, insert a gene that we want to get into, let's say, a human, and then use that virus as the delivery mechanism and let the virus do what it does, which is get that DNA into the host cell's nuclei. And how could you actually treat this? Well, this is still theoretical. They're still working on this. But there is a gene called P53, which is in all of our cells and all of our DNA. And this gene is critically important to suppressing cancer cell growth. And how it does that, we don't have to get into the details, but essentially it's involved with it's involved with killing cancer cells and preventing them from becoming cancerous. So what we've noticed then that all cancers, though they may have other genes that are mutated, they pretty much all in common have P53 silenced in some way. And so the thought process is this. Well, what if we take that gene, P53, and we take a virus, we take the viral DNA out, because we don't want that, we put that gene for P53 inside the virus, and then we infect the person with that virus. So it's going to go around and infect lots of cells. Now, if it infects a normal cell that's not cancerous, well, that cell already has a copy of P53, and so the thought is, well, it shouldn't really damage that cell because it's already got that gene, it's not going to hurt anything. But if it encounters a cancer cell and infects that cancer cell now gets a working copy of the gene, that gene does its function, hopefully, and kills the cancer cell. That's one of many examples of which we could potentially use this particular treatment protocol for. So scientists are looking at using viruses to treat cancer, using viruses to treat other genetic diseases, and so it's just one of the many ways in which the biotechnology revolution continues to roll on. So that is some real basic information about viruses. We'll get into viruses in much more detail in the immune system. For now, that's all I have for you. Hope that was helpful. I'll see you next time.